So good morning. I will do a small abstract of my work, which is done in a joint thesis between the Paris University and the Federal University from Rio de Janeiro. So nowadays, the artemisinin derivatives are the main drugs for malaria treatment. However, the substance presents some pharmacological problems, such as hydrolysis under medium acid conditions, rapid elimination, short plasmatic half-life, and poor oral bioavailability. So in our group, we developed a series of fluorinated artemisinins, and how you can see in the table, we obtained molecules with a better pharmacological profile and better activities in vitro and in vivo. So continuing this work, we designed two series of hybrid molecules where we combine two pharmacophoric groups, and the aim is not only to obtain more active molecules, but also to avoid the development of drug resistance. So we have done initial screens in vitro, and our molecules, some molecules were more active than the commercial drug dihydroartemisinin, and now we started in vivo studies of our molecules.
Next one, please.
So my work has been developed in the terms of a joint PhD between the University of Rio de Janeiro and the INES Caixin, and I'm working on the impact of an oncogenic mutation on the colon stimulation factor 1 receptor. So this receptor expression is already elevated in a number of inflammatory and cancer diseases, and this particular oncogenic mutation leads to the activating of the receptor that is characterized by big conformational change in the activation loop, and also the departure of the autoregulatory domain from the kinase loop. And this activation confers resistance to imatinib, that is the main first line treatment for cancer diseases associated with these receptor tyrosine kinases. So through molecular dynamic simulations, we are able to characterize a high amplitude motion related to the unfolding of the activation loop, and is reflected on a different pattern of secondary structure between the wild type and the mutant. But this unfolding was not correlated with the departure of the GMR domain, and this was associated with difference between the sequence of the GMR between our receptor and KIT, that is from the same family, and the more positively charged nature of the GMR in CSF1R receptor is more complementary to the negative electrostatic potential surface of the kinase loop. Nonetheless, the effect, although more subtle for CSF1R, is the same as you can see for free energy binding values compared to the other family receptors. Thank you.
Good morning, my name is Natalia. I'm a student from Unesp University in Brazil. I'm just coming from Institute Pasteur in South Korea, where I did an exchange program internship. I worked with this project that means a screening of antikinase compounds against leishmania. The methodology of the screening is already validated. We infected the human macrophages with the parasites, and then we treat with the compounds. And our plate is read by a microscope confocal, and the images are analyzed by a software that is made in-house. So with these images, we can get some data and select the best compounds in the primary screening, and we also do the secondary screening. I also did an in silico study with some software that can predict the biological behavior of these molecules. And with this data, we selected the 3 best compounds. With the 3 best compounds, I did some cellular biology study, and also we did in vivo model on mice. And so far, our conclusion is we begin with 4,000 compounds, and on the end, we have 2 compounds that are exclusively active against a mastigote form, one that is active against both forms, And the three best compounds, they are able to reduce the parasites on the liver. So thank you.
Hello, good morning. My name is Adalberto. I'm an undergraduate student also. I participated in the exchange program Institute Pasteur Korea, and I developed research about malaria disease there. So malaria is a complicated disease nowadays because there is resistance. So we need to find new targets, new methods to fight malaria disease. And my target was gametocytes. It's a specific stage from P. falciparum because this stage is really important because just this stage is able to disseminate the disease. So if you fight gametocytes, we can avoid the dissemination of malaria disease. We found a specific compound that can induce gametocytogenesis in vitro. This is the challenger because it's complicated to get gametocytes in big amount in vitro. So we found this compound and we validated this compound. We named it PP. So in this graphic we can see really clear the amount of gametocytes in culture treated with PP is higher compared to the classic methods, conditional media and dilution method. And the sexual conversion was higher also when I used PP in my culture compared to the classic methods. And here, gametocytes maturation analysis, I could see the maturation of gametocytes because there are 5 stages from 1 to 5, and I could really visualize in vitro using GIMP slides gametocytes until stage number 5. This is good for drug discovery. So in conclusion, we identified a small molecule when we could be used for drug discovery against gametocytes of P. falciparum. Thank you very much.